हेटेरोपॉलीसेकेराइड्स की बात आज करने वाले हैं. हाय एवरीवन, आई वेलकम यू ऑल टू दी अनदर ऑनलाइन लेक्चर ऑन हेटेरोपॉलीसेकेराइड्स. ऑल राइट, तो चलिए बात करते हैं कि हेटेरोपॉलीसेकेराइड्स होते क्या हैं. सो टिल नाउ वी हैव ऑलरेडी कवर्ड दी होमोपॉलीसेकेराइड्स. यू कैन सी there is a video already uploaded. So when these polysaccharides are composed of different types of sugars or their derivatives, we say that they are heteropolysaccharides, and heteropolysaccharides are also referred to as heteroglycans. Okay, so now we've covered the derivatives of sugars, and when the derivatives of sugars they join together, they are going to form the mucopolysaccharides. ओके, सो म्यूकोपॉलीसेकेराइड्स में शुगर के डेरिवेटिव्स बहुत सारे जब एक साथ जुड़ जाते हैं और पॉलीसेकेराइड बनाते हैं, सो उनको हम कहते हैं म्यूकोपॉलीसेकेराइड्स. एंड दीज म्यूकोपॉलीसेकेराइड्स दे आर commonly called as glycosaminoglycans, and we say that they are the GAGs. And now when we talk about these GAGs are also found in combination with certain proteins. All right, and when they form proteins, we say that they are the proteoglycans. And the mucoproteins or the proteoglycans, they have at least 95% of carbohydrates and 5% of protein residues. All right. So now moving on to the first example, which is the hyaluronic acid. Now this hyaluronic acid, it is the important glycosaminoglycans found in the ground substance of synovial fluid of the joints, and it is also present in the vitreous humor of eyes. Okay, so when we talk about the functions of hyaluronic acid, we already know that it is going to serve as a lubricant in the joints. वो हम कहते हैं ना कि उसके तो जॉइंट का ग्रीस खत्म हो गया है ना, तो हम जब लुब्रिकेंट की बात करते हैं, दिस hyaluronic acid, this is the important lubricant. Okay, so when we see the structure goes like the repeating disaccharide unit. Okay, we say that D-glucuronic acid and N-acetyl glucosamine, here you can see this is the structure of glucose, but at the sixth position the CH2OH is replaced by COO negative, hence it becomes the glucuronic acid. When it becomes the glucuronic acid, then of course at the anomeric carbon atom, somewhere here, okay, this is the anomeric carbon atom, and it forms the glycosidic linkage with the third uh, carbon atom of the N-acetyl glucosamine. Okay, and the linkage between glucuronic acid and N-acetyl glucosamine is beta 1,3 linkage. Now here we see we have enclosed this disaccharide in a bracket and put n number of disaccharides. So we can also say when the repeating disaccharide units, they form a polysaccharide. Okay, repeating disaccharide unit, they form a polysaccharide. Okay, so in certain mammals, in mammals, the hyaluronic acid also has beta 1,4 linkage. Okay, beta 1,4 linkage. Okay, so now this is also present around the ovum in the form of a gel, this hyaluronic acid. So when fertilization will takes place, the hyaluronidase enzyme will break the beta 1,4 linkage. So this enzyme is present in high concentration in the testes, in the seminal fluid, and in certain insects, in the venom of the insects. Okay, so when we talk about the enzyme hyaluronidase, it has important role in fertilization. Okay, because this hyaluronidase, it is going to clear the gel around the ova and helps in better penetration of the sperm into the ovum. Now moving on to the next heteropolysaccharide. So next heteropolysaccharides are your chondroitin sulfate. So chondroitin, as we see the word, it means cartilage. It means cartilage, and it is the important component of the major mammalian tissues. That includes bone, that includes cartilage, tendon, valves of the heart, skin, and cornea. So structurally, we see that it is similar to the hyaluronic acid. Okay. So chondroitin sulfate, it is made up of repeating disaccharide unit of. You can see this is the glucuronic acid, the acid at the sixth position, and this is N-acetyl galactosamine 4 sulfate. Okay, N-acetyl galactosamine 4 sulfate. So here at the fourth position there is a sulfate, and this is the structure of galactose. Why it is the structure of galactose? Because H is below the plane, and this indicates that this is the structure of galactose. Okay. So now move on to the structure of heparin. Heparin, we know, is an anticoagulant that will prevent the blood clotting. Okay, it also helps in the release of the enzyme lipoprotein lipase, which will help in clearing the turbidity of lipemic plasma. So here you can see this heparin. It contains ioduronic acid, which is 2' sulfated, and 6' sulfated and N-sulfated glucosamine, and they are together linked by beta 1,4 linkage. Okay, so now here we move on to the dermatan sulfate. Dermatan sulfate is present in the skin, the dermis of the skin, as the name indicates, and the structure is similar to the chondroitin 4 sulfate. You see, the structure is similar to the chondroitin 4 sulfate. Where is the difference? That here the COO negative, the acidic group, it lies below the plane, and when the acidic group lies below the plane, we say that it is the ioduronic acid. So the ioduronic acid and N-acetyl glucosamine 4 sulfate, they are beta 1,3 linked. Okay, and they forms the dermatan sulfate, which is important. So now moving on to the structure of keratan sulfate. It is a heterogeneous glycosaminoglycan, or the heteropolysaccharide, which contain the repeating disaccharide unit of D-galactose and N-acetyl glucosamine 6 sulfate. All right, and together these subunits, they are beta 1,4 linked with each other. So that's all for today. Do comment down that on which topic do you want the next lecture. Thank you so much. Stay tuned in.